Yeah, I welcome you all to the yet another Fusion Talk on behalf of Prescience and Insilico. A few words about Prescience and Insilico. At Prescience and Insilico, we team up with both industry and academia to tackle challenges in drug discovery and material science. The idea for Fusion Talk series it has came from our wish to get experts from different fields to share their valuable insights. And we are very proud to be one of the first Indian companies to create our own drug discovery software with AI features. Prescience in Silico was founded by academicians who wanted to make AI powered discoveries in drugs and materials accessible to everyone, aiming to redefine the landscape of scientific knowledge. And for this fusion talk, we have Professor Rituparna Samanta here. She is an assistant professor at the Department of Chemicals, Materials and Biological Engineering at University of South Florida in Tampa, Florida. Her newly formed research group develops and uses computational tools to engineer beyond soluble proteins. She worked as a postdoc at Johns Hopkins University, where her research was mainly focused on developing tools to understand proteins and their designs. Prior to this, she has earned her PhD from University of Texas at Austin, where her primary research was mainly on studying the phase behavior of self-assembled globular proteins and polymers and the effect of their physical properties on their complexes. She has spent the formative years of her career in India, where she graduated with bachelor's degree in chemical engineering from Jadavpur University and a master degree from IIT Kanpur, respectively. She has authored and co-authored several articles in renowned journals and conferences. And today she'll be discussing mostly about an implicit model-based integrated framework to advance membrane protein structure predictions and designs. With this, I hand over the stage to Professor Rituparna. And yeah, we are looking forward to the interesting talk.
Thank you, Anil, for your kind introduction, and thank you all for the invitation. I'm really happy to be here and being able to communicate and discuss science with you and learn a lot from you. So, so today I'm going to talk about, even though my title is Computational Methods to Engineer Beyond Soluble Proteins, I'll talk mostly on membrane proteins, and I'll give a brief overview of what my newly formed lab is working on. Before that, I want to walk you through where we are located. We are located in the southeastern tip of United States in the state of Florida. State of Florida is famous for many different things, but I would like to point out Disneyland, Kennedy Space Center, and the famous Keys. So you have a lot of things to visit when you are in Florida. But beside that, we have our beautiful campus of University of South Florida, located at 3 different locations. I am in Tampa campus, but there are 2 more, which are not very far away from each other. We have many different colleges. We are placed in the College of Engineering. Um, so proteins is not something which is you may all know about it already because you're working on it. So proteins are the building blocks of life, as we know, and they are built with 20 different natural amino acids placed beside each other. But how they are placed beside each other form their 3D structure, which is called the folding problem. And based on how their 3D structure is, most of the times it affects their function. But nature mostly works on evolution. However, the natural sequences of proteins are based on evolution. However, there are several problems which occurs, and that's when we have to work on design problems, where the design is to change the function of protein, what would be the sequence? Basically, the reverse problem of folding. So it is very good to see now that after several years of work on computational protein engineering, there are several applications which has now come into market. These are some of the examples, like ultra-potent vaccines from pharmaceuticals, cancer immunotherapy, and new nanomaterials. So that's really exciting. But beside the sequence, environment also plays an important role. For example, flip proteins, as shown here, at different pH and in presence of the cellular membrane, changes their structure. For example, here at higher pH, they have this an unstructured shape, and when at lower pH, it really forms a helical structure and inserts itself inside the cellular membrane. This is an example of acid proteins, or the acid-sensitive ion channels. Again, here, based on the pH, it opens and closes itself, where it allows ions to pass in, like acts as a gate. So these are important. And this is also an example of histone protein bundled in DNA based on the charge of the DNA and the protein. It forms a complex structure. So what I'm showing here is, beside the sequence, environment also plays an important role in how these proteins form a structure and how they are available in nature. This is an example of how this environment looks like. So this is a plot of different kinds of head and tail groups of eastern human proteome. So this is the variety of head and tail groups in the lipid membrane. This is mammalian plasma membrane. And this is an example of different kinds of proteins, like different kinds of polymers. Like, what can be the variety? They can be synthetic, biopolymer, their architectures can be different, their stiffness can be different, chemistry can obviously be different. So what I'm trying to tell you is, when I say that, well, the environment plays an important role, the problem is the environment is very diverse. And when you simultaneously consider the protein, which itself is a large molecule, as well as this diverse environment, it is challenging not only with experiments, it's also challenging computationally. So, and particularly with experiments, generalizing the properties and generalizing or discovering the science alone with experiments can be really difficult. So the broader goal of my lab is to work with proteins and understand how they behave in different environments. I mainly focus on proteins in cellular membranes and proteins in presence of polymers. And the broader question of the field is what kind of models. We are a computational lab. We are trying to develop computational models to understand these kind of problems. And our broader questions are, what kind of models can we develop to understand how proteins behave in different environments? And can these models help us design new proteins in different environments? So today, for this talk, I'll mostly be focusing on proteins in cellular membrane. So a brief introduction to membrane proteins. They are important for human physiology. For example, they help us smell things, so olfactory, so signal transduction in that. If you had your afternoon coffee, that's probably like a membrane protein is working to keep you awake through this talk. And then it helps in blood recognition and keeping your heart rate stable. More than that, like beside human physiology, there are also important drug targets. So about more than 60% of the FDA-approved pharmaceutical drugs are membrane proteins, even though they are only 23% of the human proteome. In spite of their importance, when you look at the protein data bank, they only constitute 2% of the structure. So there are only 2% cumulative unique structures in PDB. And that's majorly because of their difficulty in expression and purification and their structural determination. So this is honing to the fact that working or discovering membrane proteins alone with experiments can be difficult. And that's why computational methods are or can be an important tool to work with membrane proteins. So here I'll introduce our approach of which is called the implicit solvent model. So the membrane protein, so the lipid layer is formed with a hydrophilic head group or a polar head group and a hydrophobic tail group. So most of the computational methods around membrane proteins are either all atom, but with so many lipid layer and water molecule, the system becomes extremely complex. Another common way of working with membrane proteins is where these models are coarse-grained, so Martini kind of model. Even there, when the system is large, it sometimes becomes intractable. There are also several publications which talk about aggregation problems and things like that. So our approach is to replace these membrane proteins, membrane protein environment with a continuum model. So that's what we call an implicit solvent or implicit membrane model. But we represent the protein with all atom details. So we lie somewhere in the middle, where middle of coarse grain and all atom, where we do not have an all atom representation of the lipid membrane. So we replace that with a continuum function, but we stick to the all atom representation of the protein. This way, we save some time and publication, and there are previous literature which has shown that implicit membrane models can can speed up your simulations by about 50 to 100 times. But there is a challenge. Adding realistic features of the membrane environment and membrane environment is the biggest challenge. So our objective here is to add realistic features to membrane environments, to these implicit membrane models, and evaluate their ability to capture experimental observations. So we'll be using Monte Carlo methods for most of the simulations we have done. So that relative to molecular dynamics, which talks about time progression, we can change steps, and we don't have to go through the time progression. And that way we think like Monte Carlo can reach the equilibrium state. Our objective is to reach the equilibrium state. So Monte Carlo relative to molecular dynamics can speed up that application. So that way also we are a little faster. The way we approach how do we so the way we approach is we take an iterative step. We do some changes in the model, and then we have test cases. So we have collected experimental measurements of membrane proteins relative to, say, orientation, stability, structure, and design. And so we have a 12 independent test set. So we do some changes in our implicit membrane model. We test it through our benchmark suite. That's what we call it. And then we analyze the results, and then we see where the difference is, and then we come back and redevelop. So we take an iterative approach to build the model and test it and then apply it to translational research. So today I'll show you one round of this iteration, and then I'll also show its application in terms of docking. So this is an example where, so when I joined the lab, there was this energy function for membrane proteins. So energy function is basically, because we are doing Monte Carlo, so we need it on the need a formula to calculate energy. So energy function is that formula to calculate energy of a membrane protein in this environment. So there was already an energy function, which is called the Franklin 2019 for membrane proteins. So we first tested it on how to calculate the tilt angle. So tilt angle is this equilibrium angle which a protein makes with the membrane. So the angle which it makes with the normal z-axis is called the tilt angle, and the depth at which its center is from the center of the membrane is called the depth. So we are trying, so there are experimentally measured values of this. We are trying to predict them with our energy function and then match it with the experimental value. The way we do it is we take the peptide backbone, and then we move it in all sorts. So we rotate it around this axis. We take all, like, we tilt it through all the angles of the protein and also of the membrane. So we do like a tilt angle sweep basically, and a sweep in on the depth as well, and then make a energy landscape, and then we pick the minimum energy point. So here the blue one shows the tilt angle calculated by Franklin 2019, and NMR is shown here in gray. So experimentally, the peptide is shown to be on top of the membrane surface, whereas Franklin 2019 predicts to be inside the membrane. So this was one of the results which where Franklin 2019 did not perform well. So we are here to analyze it. So this is the energy landscape I was talking about. So on the y-axis, we have membrane depth, and on the x-axis, we have angle, and all these points are minimized over its rotational axis. So minimum energy at a particular angle. And here, the NMR value is shown here in triangle, and the white dot shows F19. IMM1 is one more previous implicit membrane model. So this was what the landscape was at that point. None of them were close to the experimental value. Franklin 2019 was a little better than IMM1. So let us see how Franklin 2019 was different. So the first, the IMM1 model assumed empirical implicit water density model, which was fixed, whereas Franklin 19 had a library of lipid membrane types and could change the water density, had a, like, the water density was different based on the lipid membranes. So that was one thing which was an improvement. The other was the transfer energy for residues for IMM1 was based on water to an organic solvent transfer energy. However, for lipid membranes, like the cellular membrane is a little more complicated than representing it with organic solvents. So for Franklin 2019, the transfer energy function was taken from experimental values of residues folded into like a beta barrel. So these were experimental values taken from the Fleming lab. Also the pores. So the membrane proteins have different kinds of pores. For IMM1, it was assumed to be a cylindrical value, whereas for Franklin 2019, the values of the pores were different for different z directions. So that was also considered. However, so mathematically, what the extra energy function was, it took care of the changing density of water inside the membrane as well as the pores. And this was the result which we got. So if we look closely upon analysis, what we found was there were flanking lysine residues in this tail of this peptide. And so these lysine molecules, because they were polar, Franklin 2019 pushed it outside the membrane and put the peptide backbone inside. So what we realized was these head groups, which are also charged, we don't have any interaction between these residues with the head group. So the electrostatic interaction, because of this membrane, was missing in this energy function. And there were a few more things. So once we could include that, we also realized that because the water density inside the membrane is different, so the dielectric constant should also be different. So in the previous version, Franklin 2019, the dielectric constant was same as that in the center of the membrane and outside the membrane. So that also we realized we could change. So basically this part of the talk is how we can add the electrostatic features of these membranes. So how we did it is we did molecular dynamics like simulations for only lipid and water, and then we extracted the electrostatic potential for these lipid membranes. We also included, based on the water density, we had formulas where we could include the changing the changing dielectric constant inside the membrane. So here is the membrane center, and here is the distance. So based on from the soluble region to the membrane center, the electrostatic, so the dielectric constant would be different. So that also we changed. And so now after, so the first part is the soluble energy, like we had in Franklin 19. Then we had the Franklin 2019 transfer energy function. And then we added two more terms where we had the change due to the dielectric constant and also the interaction with the lipid head group. So now it's again, so like I said, it's an iterative process. We did some changes to our energy function. We would now test the tilt angle calculation. So this is what we had with Franklin 19. And now the pink one is the calculated Franklin 2023, super close to the NMR results. So this was the result for Franklin 19. Now we are here where the results are close to the NMR calculations. But is the tilt angle, so did Franklin 2023 improve the tilt angle calculation on the fly, or was it really due to the electrostatic terms? So here we show the per residue energy for Franklin 2023, 19, and average position, and they are mostly due to those lysine residues like we hypothesized. But if you look at the results, there is still difference between the experimental result and the predicted value. One thing which we think, one thing which we missed is in this version, we did not change the backbone of the peptide. So these are small peptides and they are really flexible. So these are all the NMR structures of this peptide. We just took the lowest energy structure and just and did a sweep in the tilt angle and the depth. But I think if we incorporate the change in the backbone movement and then average the tilt angle, I'm sure we can. We hypothesize that we can improve that tilt angle prediction further. Next question is this observation universal? Did 2023 only improve the tilt angle calculation, or could it also improve anything else? So we tested it on 12 cases again, and relative to Franklin 2019, Franklin 2023 outperformed on 8 out of 12 cases. Here we tested them on like tilt angle of peptides, tested them on stability, and also design problems. So in effect, we made a tool. We have a platform to look at membrane proteins, and we have a tool set here where we can test them on orientation, stability, docking, and design. So I'll go ahead and show you an application of Franklin 2023 on docking. So here I show a bound complex, meaning two proteins are together, and this is a crystal structure of mu opioid receptor. And they are made up of two proteins. So these are the unbound monomers. And when we overlap the unbound monomers on the bound complex, you can see that there is a difference between the two. What I mean by that is when these monomers come closer to each other, they wiggle in presence of each other and then forms a complex. So can we capture that? And when we were working on this, AlphaFold multimer came, which is like, I'm sure you all know about this, that you just have to input the sequence. This is a deep learning based protocol which takes in the input of the sequence and predicts the complex structure. I can see chats over here. Um, can someone read out the questions if it is there, or shall we take it after the
Yeah, we'll
talk?
be taking those questions at the end of the talk.
Okay, okay,
Yeah.
sure. Then I can go ahead. So this is, so we took, so we questioned ourselves. So how does AlphaFold multimer work on these membrane protein sequences? So we gave the sequence. The gray is the crystal structure, and the pink is the prediction of AlphaFold multimer. And these are basically two, like the front view and the top view. Uh, and you can see that the pink and the gray doesn't overlay on each other. So the prediction is not that great. We made our physics-based models. So this is our prediction where at least the interface overlays on each other. So we did better. So I'll tell you what were our tools, how did we do differently. So let me tell you why understanding complexes are important. So oligomerization of membrane proteins on cell surfaces play a fundamental role in several applications, such as glucose transporters. They change conformations in like mysins, and also like receptor tyrosine kinase, where changes in the transmembrane domain or mutation in any other part lead to diseases. So despite their significance, modeling these can be difficult for the reasons we discussed before. So diversity of the environment, the membrane protein structures are really underrepresented, and also like we are doing an implicit membrane, but if we have to represent these complexes as well as the lipid environment, considering them can be computationally challenging. So this 2% structures is only for, remember, they are only for membrane, like single structures, so membrane protein monomers. We are talking about complexes. So we found, I'll show you later that we could only find out 30 complexes where we know the bound and the unbound structures. So so the the best method which we could find out for membrane protein docking at that point was Jabberdock, which uses Olata molecular dynamics to first equilibrate the system, and then they use shape complementarity method to find out the interface. And the challenge which we figured out where the system was extremely expensive and can pose difficulty for large proteins. And so our objective was to try membrane protein docking with our implicit membrane model. So we are again using Monte Carlo for this. So we have the structure of proteins. We use backbone movements, and then we score it and do this iteratively. So we not only change the backbone, we also translate them, rotate them at different positions relative to the other, relative to each other, and then see what is the energy. And like we iteratively do this again and again till we reach the lowest scoring decoy, or the lowest energy position. Uh, so we also will show you how we have included machine learning tools uh, in uh, in addition to this traditional method. So we will show you how we have actually made a hybrid method. So we are talking about movement of the backbone in presence of each other. So we originally had a rigid docking protocol where we would take the structure of proteins of the monomers and then translate and rotate it relative to each other alone. That is called the rigid docking. But now what we are showing is ensemble docking, which means because to capture the change in backbone relative to each other, we also create ensembles of these two monomers and then do the translational and rotation relative to each other. So besides the rigid docking methodology, we also have changes in backbone and then docking. So this is our result for rigid docking. So we have two cases shown here. The gray is, again, the bound complex. The unbound structures are blue and green. So this is a rigid, so when the root mean square deviation between the unbound monomer and the bound complexes are low, then that means we call that a rigid target, because that means they have not moved so much relative to each other. Whereas for a flexible target where the monomers have moved relatively higher, so then the root mean square deviation between the bound and the unbound would be higher. So on my right, I have a flexible domain, and on my left, I have a rigid structure. So with rigid docking, the rigid structure prediction is really good. How I say that, here I plot on y-axis the interface score, and on x-axis the interface root mean square deviation between the bound and the predicted structures. The blue is the bound result. So when the dots are the predictions. So if the dots match the blue stars, then I have a good prediction. So here I do have the dots matching with the blue stars. So I have good prediction, which is close to the bound structure. Whereas for the flexible one, the dots did not come closer to the blue one. That means my predictions are far away from my experiments. When we have ensemble docking, on the other hand, with the rigid docking, we are still good. But with the flexible docking, we could come closer to the rigid docking, much better than the rigid docking. We didn't do the experiment on only one structure. Like I said before, we could collect a benchmark of about 30 structures where we had both unbound and the bound structures from PDB. So these are the results from Jabberdock. So this is the best IRMSD, so meaning the lowest IRMSD structure on the y-axis, on the x-axis at different protein complexes. This is the rigid docking results, and these are the ensemble docking results. So the blue one are our new model. So consistently for most of the structures, we have done much better than Jabberdock with less resource used and faster results. We also asked whether, so by then AlphaFold 3 came in. So we thought, okay, let us check that. And these are the results for AlphaFold multimer and AlphaFold 3. The pink structures here are models represented. So the gray is the experimental result, and green, the teal color is their best predicted structure. And the pink, so they produce about 5 structures and they rank it. So the blue one is the best ranked structure and the pink one are the rest. So if you see, they create structures which are not close to the experimental bound complex, whereas our method could predict the interface correctly, and it is closer to the experimental complex. So I'll show you what we did with the hybrid method. With the hybrid method, what we did was we first predict the complex with AlphaFold multimer, and then use our ensemble docking on top. So our starting structure, instead of monomers kept at like random distances and angles. We start from the prediction of alpha-fold multimer and then use our ensemble docking method. And then, so alpha-fold multimer are the gray ones. And when we use rigid and ensemble docking, you can see that for most of the, almost all of the cases, the RMSD, which means the difference between the bound and the predicted structures, are lesser. And you can see it better here where I predicted like the IRMS or the interface RMS of alpha-fold multimer and alpha-fold multimer with ensemble docking. And for most of the cases, it is either low or almost same to each other. So that was one application. The other application, which I'm not going to talk, but we are still working on it, is how can we design these beta barrel structures, which can place itself inside the cellular membrane. So here I show you a soluble membrane protein, which also has this cylindrical structure. They are present in these fluorescent jellyfish, and similarly, a similar looking structure, also cylindrical in shape, present itself in the cellular membrane. One is stable and one is not so stable. So how can we design stable membrane proteins is the question. We are still working on it. So in conclusion, I showed you for how we are iteratively designing implicit membrane protein model. We showed that Rosetta MP Doc, the application project which I showed, successfully predicts and corrects interfaces for about 67% of moderately flexible and 60% for highly flexible targets. However, Rosetta MP Doc still fails to capture large conformational changes, and we think that needs better scoring and better conformational sampling. We did better than previous cases, but we still need to do more. All the protocols we develop are accessible through Rosetta Commons software. It's an open source software, but we also try to develop like tools or platforms or web-based platforms where people can just submit their sequences and can get the complex. So we actually developed, so Rosetta MP Doc starts from a structure, but with AlphaFold 3 or AlphaFold Multimer, we combined and made a tool where even if you don't have a structure, you can predict the structure through AlphaFold Multimer and then use our hybrid protocol for that. Time is moving so fast. I have been in this job for just 7 months, but the lab has grown. I have a couple of master's and undergraduate students and a few incoming graduate students. Most of the work which I showed you today were done during my postdoc, so a great shout out to Gray Lab. So the application project was in collaboration with a graduate student, Amaya, and an undergraduate, Priya. And we have our code reviewer in Rosetta, so thanks to them as well. The funding sources, NIH, and our computational resources come from like Arch and CERSI at USF. And thank you all for your attention. I would love to take any questions and comments and learn a lot from you.
Yeah. Yeah, thank you, Professor Rituparna. It was a wonderful talk. Yeah, so there are questions coming up, and I'll take one by one. So the first question is asked by Steve. He asked energy function for the interaction energy between the head group of lipids. Is this function dependent on the distance between protein, let's say a center of mass of protein, and the position of the head groups?
So with the span file and everything, we place the protein in the membrane protein, and we have our electrostatic potential as a distance of the membrane protein height.
Okay.
So based on where the membrane protein is, we calculate the energy based on that.
Okay. Another question is from Sudeep again. So, is there an AlphaFold model for membrane proteins only? Let's say the 2% proteins you were, talking about. Is that available?
Oh, so the structures for membrane protein, the protein data, so AlphaFold 2, not the 3. The AlphaFold 2 actually created like structures for all the sequences available in UniProt. So there are structures for membrane proteins as well, but it is not very clear whether those structures were there in their training set or not. So that we don't know. The structures, the complex structures which I showed you, wait, so the question is that 2% structure, the question is about the 2%
No,
I
the
showed,
question is
the
about,
structure.
yeah. It's about the AlphaFold model for membrane proteins. Is that available?
Oh, I'm sure it is available because AlphaFold 2 created a database for all the sequences available in UniProt. So uh, I, I I think they're available from that AlphaFold database.
Okay. The next question is from Simon. What would be your strategy to model transportable channels that breathe
It's
and
not very
changes?
clear.
Okay.
I can't.
So what, can you hear me?
Yes.
Yeah. So what would be your strategy to model transmembrane channels that breathe and changes conformation upon solute transport across them?
Yeah, so that's a very good question. We can't handle it at this point because remember we don't have solvent here, or we don't have explicit ions here. That is something which is which is my future goal in the lab to think about, even like uh, can we and I don't have an answer for this right now. So can we add these solute transports being in an implicit model? I don't know. And particularly because we are I'm not interested in dynamics or transport yet. But that's a question which has been hovering in my brain for a long time, even for static systems. So you were asking about a dynamic system, which we are not interested in, or I would say our system cannot handle it. We mostly look at equilibrium structures. But even in equilibrium structures, if I have a parallel question, how can I handle solute and solvent? We can't at this point, but that's something which we would like to take up.
Yeah, I had one question. So this is regarding the Jabber dock. I mean, the docking of the transmembrane protein. So how is it different from, let's say, a charm GUI setup? I mean, where they have this membrane builder where you can have protein, at the center, followed by setting up lipids around it. So how is it different from docking? Why would I go for
No, so
docking?
Jabberdock actually used the traditional
Okay,
molecular dynamics. So I forgot if they used charm GUI or something else, but they used a classical, like traditional molecular dynamics to equilibrate the structure, like equilibrate the protein structure around the lipid membrane. Once they equilibrated it, then they used shape complementarity. So they did not do the whole equilibration with
Mhm.
traditional traditional molecular dynamics. And these are strategies to like move it faster, and then because it would take like a really long time to reach there.
Mhm. And yeah, so what was the force field you have used for, let's say, to model protein and
So we do, because we use Monte Carlo, we don't
oh,
use any force field. So
energy?
parallel of force field is the energy function. So
Right.
that's what we developed.
Okay. Okay. So that was from my side. If there are, yeah. So there is one more question came from Sujit. So yeah, he asked how the tilt angle and the depth of peptide chain changes with the ratio of hydrophobic is to hydrophilic residues of membrane protein.
That's a good question, but we didn't analyze like the tilt angle that way. I mean, this is a question which we can answer obviously with our setup, but what we did was we took about, I think, 30 to 40 tilt angle cases experimentally found out, and then just predicted the tilt angle. This is a very good question. I don't have an answer for that, but we can analyze that if uh, if needed, we can analyze that. We have the tool set to do it. But this was a test more on can we predict it or not kind of a situation.
Yeah, so if there are no more questions, I would like to ask Dr. Sudeep Roy to give a vote of thanks.
Yeah, thanks, Anil. Thank you. Yeah, thank you, Professor Samanta. So it's very nice talk, and it's very interesting. Means I have been following the membrane protein literature for some time. I've seen some molecular dynamics, nice molecular dynamics, even done by here in IISc by in chemical engineering department. So, and this is a very nice take on that, right? Means how we can speed up the method so that we can do large membrane protein assemblies as well. And the future goal you have just talked about that, how you can incorporate solvents and ions into the channel. So that will be like a, final goal for it, right? So there are so many membrane proteins. And we need to understand that from the drug designing and drug delivery perspectives as well. And that's really lacking currently. So it's really thank you for coming and giving a talk. Although we would love to have you hosted at Prescience, and if you're coming sometime in Bangalore, please let us know.
Yeah, I
We'll
would
be
love
happy
to visit you as well. I just came back from India, unfortunately. So maybe next time.
Yeah, next time, please. Yes, please. Yeah, I see that Jayant was also there in the meeting. Jayant, maybe you know from your IIT Kanpur days.
Yeah,
So he's
absolutely.
yeah.
I mean, he was my professor. He was my first teacher of statistical thermodynamics. So I learned a lot
Yes.
from him.
Yeah. I'm not sure whether he is still there or not. This is one of our effort to have a nice discussion around the world, what is happening, what is there any cool science happening, how people are doing new things. And we try to learn from it. And not only we from the company, we make it open to all. So it's basically anybody can
That is really
register.
great.
And
Yeah.
that's and so Rituparna, for that actually means, this is actually it's a network growing slowly. And we would love to see you again coming and giving a talk about on your vision that, after a year or so when the work will, really come to some stage. Yeah. So other than that, I would like to thank Anil for organizing the event, as well as Meghna for helping taking the word out. So we generally post it through LinkedIn, post it through several other channels, and let know people that we are having a talk every month. We are having that. And of course, the IT team for Zoom links and sharing everything with us, and on all the audience staying together for almost 50 minutes now. And thank you very much. Thank you again.
Thank you so much for very interesting questions.
Yeah, and it's a Zoom, so we face to face becomes really very nice.
Yeah, so thank you everyone for joining. And again, once again, thank you, Professor Rituparna Samanta.
Yeah, thank you so much
delivering this talk.
Yeah.
Okay.
Bye.
Thank you.
Bye-bye.
Bye-bye.
Yeah.